Right. Good afternoon, everybody. I think we still have a few people trickling in, but as we get started, I'll just introduce the event and get us going. So my name is Tefera Jemian. I'm the Communications Officer here at BioCanRx. I'd like to give a special thank you to the National Research Council for collaborating with us on this event. We're very happy to have you, and we're very happy for this to be happening. Just a second here. Oh my God, sorry. Give me just a second here to get a slide worked out, and we'll get started here. So as we begin, we'd like to acknowledge that BioCanRx's work unfolds across Canada on the traditional and unceded territories of Indigenous peoples and nations who have stewarded these lands for millennia. We respectfully acknowledge the Algonquin Anishinaabe Nation, on whose land our head office is situated, and we extend this recognition to Indigenous communities across the country. We honor their enduring stewardship of the lands and waters where our researchers, partners, and communities live and work. As a national network, BioCanRx embraces the responsibility of advancing reconciliation by embedding respect, inclusivity, and Indigenous leadership into all aspects of our mission. So with that said, I'll just say thank you very much to everybody for joining us today to talk about the great work in cell and gene therapy being done in Canada through the NRC CGT program, and I will hand it over to Rizini to get us going.
All right, thank you very much, Teffer, and again, thank you to everybody. Good afternoon and welcome. So today we will present to you the work that we've done through the Cell and Gene Therapy Challenge Program at the National Research Council. As Teffer mentioned, the webinar is titled Driving Innovation in Cell and Gene Therapy, Collaborative Discovery and Patient Impact through the NRC Cell and Gene Therapy Program. So I'm sure everybody here will agree with me that cell and gene therapies are rapidly transforming medicines. They offer the possibility of treating and in some cases curing diseases that were once considered intractable. So, but turning these scientific breakthroughs into real therapies for patients really require more than discovery. It requires right infrastructure, right expertise, strong collaborations across many sectors, academia, industry, healthcare, and government. So over the past 7 years, NRC Cell and Gene Therapy Program has been working with partners across Canada to really try and help build a foundation for a homegrown biotherapeutics ecosystem. So together, we have helped enable the first clinical trial of a truly made in Canada CAR-T therapy, which you will hear about today at this event, and also our efforts to take these therapies to the next stage of treating solid tumor cancers, and also our efforts in developing off-the-shelf cell therapies. In addition, we have also helped advance scalable manufacturing approaches for viral vectors and developed precision delivery technologies such as targeted lipid nanoparticles for really tissue or cell-specific delivery of therapeutic cargo, which you will also hear about today. We have also expanded Canadian infrastructure for GMP production of CAR-T and viral vectors to help clinical development of these therapies that we are building together or developing together. And in addition, we've also supported the development of foundational resources, in particular the high throughput mammalian gene editing capabilities at Concordia University. For that, we've developed a number of genome editing tools, as well as a research bank of induced pluripotent stem cells derived from human amniotic fluid cells, really to help accelerate discovery and therapeutic development. So unfortunately today, we don't really have the time to talk about the genome editing tools and the iPSCs that we've developed, but we will be having a focus session on those technology platforms at the International Society for Stem Cell Research meeting in July, which is going to be in Montreal. So if you are there, we will be hosting an innovation showcase session where we will be talking about these genome editing tools and the iPSC technologies. So what makes our collective work through the CGT program particularly exciting is that these capabilities that we've developed are not just for scientific achievements. These technologies and platforms are really designed to be used to innovate in the space of cell and gene therapies and to make new medicines for Canada and beyond. So these technology platforms are available to Canadian researchers, companies, and innovators who are working to bring the next generation cell and gene therapies to patients. So today's webinar is really an opportunity for us to showcase these capabilities that we have built and to highlight how collaboration with NRC can really help bring these promising ideas from concept to patient impact. So we are really pleased to have an outstanding group of speakers and moderators with us today who will be sharing their perspective and how these platforms are being applied and how they can support innovation across the Canadian ecosystem. So with that, I'd like to introduce today's moderators. For the first session, Dr. Scott McComb is a senior research officer with the NRC's Human Health Therapeutics Research Center. He's part of the Cancer Immunology Group. He's also an adjunct professor with University of Ottawa. Dr. McComb led a flagship project of the cell and gene therapy program, Made in Canada CAR-T for hematologic malignancies. For that, he really drove the collaborations both internally within NRC as well as collaborations across Canada. And in doing so, he has really built a very robust tool set to design and develop CAR-T therapies, and he's really adding to these tools every day. And these tools will be available to clinicians and researchers across Canada to really build the next generation safe and effective, affordable and accessible cancer treatments. So he will moderate the first session, which is on engineered cell therapies. And the second session will be moderated by Dr. Umar Iqbal. I hope, Umar, you're online. It would be great if you could put your camera on if you're online. Dr. Umar Iqbal is also a senior research officer with the National Research Council, Human Health Therapeutics Research Center, and he's focusing on advancing innovative therapeutics from concept to preclinical validation, spanning antibody-based drugs, lipid nanoparticle platforms, and gene regulation. His work is driven through multidisciplinary collaborations across academia, biotech, and pharma, with the goal of translating discovery into real-world impact. And most recently, through the cell and gene therapy program, Dr. Iqbal led the development of proprietary targeted lipid nanoparticle systems engineered to efficiently deliver therapeutic cargoes to the brain, hematopoietic stem cells, and T cells, overcoming key barriers in cell gene transfer and enabling new possibilities in gene and cell therapy. So thank you again. Thank you to BioCanRx for helping us put together this webinar. And I will now hand it over to Scott for the first session.
Okay, thanks so much for seeing that great introduction. And I'm just going to share my screen here. And maybe Teffer, you can just give me a thumbs up. You can see the screen.
Looks good.
Yes.
Yep.
Okay, thank you. All right, so it's certainly my pleasure to be here today. My name's Scott McComb, as Rossini said. I had the honor of leading the Cell and Gene Therapy Challenge Program Master Project 2, which was to develop blood cancer CAR T products and to try to bring that to clinical trials. So I have some slides to introduce the topic of CAR T and where we began with the cell and gene therapy program back in 2017 and where we are today. So it's really a story of taking CARs and bringing them from an idea all the way to practice or to a clinical application for patients. I have a picture here of a llama in a car, so hopefully that'll make sense why we have a llama driving a car by a few slides to my presentation here. So, of course, we've known for a long time that immune cells are able to kill cancer cells. This is a well-established fact in cancer research. And of course, sometimes immune cells can kill cancer cells, but by the fact that cancer occurs, we know that cancer cells can escape the immune system. And we've for a long time never fully understood why cancers are able to escape immune function. But we've had a dream that if we could take those immune cells and reprogram them with genetic technology in order to allow them to recognize the cells, we could make changes, reprogram the cells, and allow them to recognize and kill all of the cancer cells. And this is really the science fiction type dream, I think, that is CAR T technology. And this is done through use of chimeric antigen receptors that are introduced using gene transfer technology, modified viruses that have been changed to be able to only deliver a gene and no longer infect or cause disease in the cells. The genes go in and then induce creation of a chimeric molecule. Chimeric means a combination. So it has an external part that looks like an antibody. This has ability to grab onto certain target proteins, and it has an internal part that has signaling. And so when the external antibody part of the chimeric antigen receptor recognizes a receptor, such as that expressed on a B-cell cancer, it grabs on. This activates the signaling module of the CAR, and then this signals the CAR to do things. And one of the things that it can do is to release toxic mediators that can kill the cancer cell. And then in addition to this, not only do the cells kill, but they also copy themselves. And so these are the two critical functions of a CAR T that when you infuse these cells, they're able to go throughout the body, find the cancer cells, and upon recognition, copy themselves to create more cells that can kill more cancer. And this, sounds nice in theory, but this is what it looks like when we do it in the dish. Here you can see green cells. These are CAR T cells. We've modified human T cells genetically in order to allow them to recognize here. These are ovarian cancer cells. And you'll see when I activate this, you can see the T cells grabbing onto the cancer cells and actually killing them very rapidly. And so this is what it looks like in the dish, but actually it's even more amazing when you see it in the real world. This is from a news article from 2017 talking about the use of CAR T cells for multiple myeloma. Multiple myeloma can occur throughout the body. It's a disease of the bone marrow, but you also see it occurring throughout the body. Each of these dark patches is PET imaging showing cancer throughout the body. After CAR T treatment, you can see the CAR T removes the cancer, kills the cancer throughout the body. And this is really miraculous. And this is what it looks like in a scan, but when it's a patient, it's the difference, about being able to live a full life. Two patient advocates here who have been treated with a CD19 CAR for lymphoma in Canada on the CLIC-01 trial, which was a really innovative, the first trial of a made-in-Canada CAR T product. And both of these patient advocates now have become really strong advocates for CAR T because it's really given them hope, and through this treatment they've been able to survive despite the fact that other therapies were not effective for them. So this is the dream that we really want to realize for more patients in Canada. So that was the part of the idea of the Cell and Gene Therapy Challenge program. So back in 2017, there actually was only 4 trials ongoing in Canada for CAR T, whereas you can see that there was many trials already occurring in the USA and China in particular, but really a lot of international trials happening. And even these 4 trials in Canada were all additional international arms of US-based CAR T trials. We had very limited capacity to develop our own CAR T products. So this was part of the problem statement at the beginning of the CGT program, was that we had, despite the fact that CAR Ts were developing rapidly internationally, even as of 2017, it was clear that regulatory approval was on the horizon. Large phase 3 trials or larger trials were happening, but Canada had limited capacity to develop our own homegrown CAR Ts, especially novel CAR Ts that were against different targets than those that were already being used or for different types of cancer. In 2017, there had actually been no trials of a Canadian manufactured CAR T products. And so this was part of something NRC wanted to try to participate, to stimulate and catalyze the development of CAR T therapies because of the transformative power of this technology. We wanted to enable the development of Canadian solutions to improve accessibility and affordability of engineered cell therapies. And I think we really accomplished a lot of that in this project, and hopefully I can demonstrate that as we talk about what we really did. And so coming to this Made in Canada to CAR T project, in 2017, there were several targets that were known to work well. As I already mentioned, CD19 CAR Ts were quite advanced, but there was other targets that were very similar to CD19. They were expressed on blood cell cancers and were known to be able to be targeted by CAR Ts, but were less advanced as far as current generation therapies. And so we targeted actually all three of these. So there's BCMA, CD22, and CD19, all expressed on different forms of B-cell cancers. And we decided we would make antibodies against these. Now, not only would we make antibodies that could be used for CAR T, but we decided to focus on a particular type of antibody that occurs in llamas. Llamas, camels, and other types of camelid species are known to have what are called single domain antibodies. So to explain this, you have to think about in humans and mice, rabbits, most mammalian species, they have antibodies that have two chains. So they have two parts that actually grab onto the target antigen. And you need both a heavy and a light chain to come together in order to be functional as an antibody. This is different in llamas and camelids, where they are able to function with just one chain. And so these heavy chain only antibodies actually can be highly functional. And this allows us to take a single gene and create very rapidly new CARs and to find those functional CARs much more rapidly. And so this initial antibody discovery work, which involved producing large amounts of CD22 antigen, for example, injecting a llama with that vaccine. The llama will raise an immune response against it. Then we will identify those single domain antibodies that are able to bind to that antigen using phage display technology. This work was led by a senior scientist here at the National Research Council, Dr. Mediar Babi-Garudi, and really he was able to identify antibodies against many different parts of this large CD22 protein. At that point, we had very good antibodies we knew could bind CD22 expressing cells, but we didn't know which would be good for a CAR. So we had to take each of these different antibodies and screen them in some way. And in 2017, we didn't have an established process yet on how to do this. So we actually created modular CAR plasmids. So this is a container for the chimeric antigen receptor where we can rapidly just swap out the different parts of the CAR, and in particular the antibody binding portion, and then we can screen those very rapidly. And this has become actually a very useful tool, not only for us, but throughout the ecosystem. So we've actually shared these through the AdGene repository with more than 385 institutes around the world. So I think this has been really a good success of disseminating useful scientific tools, not only within Canada, but internationally. So taking these modularized CD22 CARs, we screened them, and here you can see we looked at whether the T cells that express these CARs actually become activated. So those that are activated will have higher expression of this marker called CD69, and you can see here when we express them on T cells, many of these CD22 targeted CARs activate, but not all of them. and that's the higher bars here. But if we remove the CD22 antigen specifically from those, we see no response. And so we were able to get a nice list of CD22 specific antibodies, where about half of those were able to respond to CD22 in an antigen specific manner. And we went through a lot of additional in vitro work in the cell, in the dish, in order to identify those that had a really powerful response against the CD22 antigen. And here I have a video. Hopefully this comes through well. On the left, you see our CD22 specific single domain CAR. Green cells growing very rapidly and killing all of the tumor in the dish. On the right, we have the same CD22 CAR, but our target cell, we've removed using CRISPR technology, the CD22 antigen. And there you see the tumor is not recognized by the CAR T cells, and the tumor, the red cells, continue to grow unabated. And so here we've had produced a CD22 specific CAR that could respond very rapidly. We produced some interesting biological insights that recapitulated what others have observed, in that the single domain antibodies need to be relatively close to the membrane. You have to actually bring the CAR T cells and the target cells close together in order to get them to really be functional. And so we published a paper on this, and if you're interested, I encourage you to look it up. It was published in Molecular Therapy Oncology, and tells the whole story of how these CD22 single domains were created and screened. And at this point, we started working with the Canadian-led immunotherapy consortium. This was an established group of researchers from BC and Ottawa that worked together to create the first CD19 CAR T, and we worked with them in order to take our CD22 single domain antibody and create a clinical version of that could be used for a clinical trial. The plasmid production, the initial CAR design, was led by Rob Holt. Jennifer Queasy created the GMP lentiviral vector. Scientists in Victoria, initially led by John Webb and later Julie Nielsen, created the CAR T manufacturing process. And Kevin Hay and Natasha Kekri worked together to create a clinical trial design and have taken this into clinical development. And so this was actually one opportunity where we had an issue. For what I showed in the video, we're using actually a trick to identify those CAR T expressing cells. It's based on fluorescent protein, but if we don't have that, how do we actually see? So we created an antibody against the single domain portion of the CAR, and this was a critical tool for allowing us to quantify and qualify CAR T products using single domain antibodies. And these antibodies are now available through an antibody distributor called Absolute Antibodies. And these are actually very useful tools, and there's an interesting paper also just on the development of those tools. And so now when we made this preclinical version of the CD22 CAR, we did additional studies working collaboratively with the CLIC group, and found that we're able to, in a mouse model, actually almost completely clear lymphoma, and the mice lived a very long time after treatment. And this was combined with many other studies. We needed to confirm that the specificity of the CD22 was consistent when we looked at it in the diversity of the types of tissues it would see if used in a living human. And so we actually, again, used our single domain antibody technology in order to look at where these novel antibodies, the novel anti-CD22 single domain, binds either in B-cell rich tissues where we did see binding as expected, or in other tissues where we don't want the CAR T to activate, such as brain, skin, or kidney. And so we saw minimal binding outside of lymphoid tissues, and this encouraged us to proceed with clinical trial plans. And I'm very happy to say that the clinical trial is now ongoing. This involves an incredible number of people across the country. Cells are actually flying from different clinical sites in Toronto or Ottawa, Calgary, to a manufacturing site in Victoria where the CAR T is created using Miltenyi Prodigy. And then those CAR T cells are returned and are infused in patients. And at this point, I can say more than 10 patients have been treated, including pediatric patients at Sick Kids Hospital. Of course, this time we don't have any information on the efficacy, but we do expect clinical trial results sometime next year. So it's a very exciting time for this therapy. And so I think between 2017 and 2026, NRC has done its part to try to stimulate really impressive development of this technology. CD19 now is standard therapy in Canada. Demand is growing because CAR T is more common. More people are also relapsing after initial CAR T, and new CAR Ts could be useful in that post-CAR T relapse cohort. Canada has robust science, said all the time. We have a collaborative culture. We can do more, but we need to take it beyond the science lab. And so CD22, the single domain CAR program project was supposed to serve as a flagship. I think it did that. It helped to establish R&D, manufacturing, clinical know-how, and push towards this product of Canada CAR T. We're now working on development of solid tumor CAR Ts, and Dr. Nelson is going to give us a very nice introduction to the development of a solid tumor CAR T. And then we're also working on development of off the shelf to try to scale this. And Dr. Lee in the next talk will, after Dr. Nelson, will give us an introduction to CAR NK. And we're also working with commercial entities to try to deploy this across Canada in a wider fashion and globally. So I in one more minute, just talk about we have gone beyond CAR. So of course I talked about how we can reprogram T cells. We'll talk about CAR, how we can reprogram NK cells, but we can actually take this same nanobody technology and create immune engagers that we could inject protein and get very similar responses. And maybe there's advantage to having both of those technologies. And there's places where both can work as treatments for cancer. And so we're developing those studies now, experiments in order to compare the same antibody in different contexts. And of course, we have many targets where we develop nanobodies. And so I've given a rundown here. We have many blood cancer CARs where we are interested to develop new therapies for cancer and for autoimmunity. So if anyone here listening to the webinar is interested, I encourage you to reach out to me. Solid tumor, again, we have many targets that we're interested to develop new CARs, new immunotherapies, and to go beyond blood cancers. And then finally, I think there's so many advantages to off-the-shelf. CAR NK is very compelling. We'll hear more about that from Dr. Lee. So, of course, I don't have time to go through all the acknowledgements. It's involved many, many people at NRC, outside of NRC, more than even I think I could fit on one slide. Hopefully that comes across. It's been an honor to be able to work on this, and I think the future is really bright for this, and I think Canada's really just still getting started in how much impact we can have on treatment of cancer and other diseases with cell therapy technology. And so I'll just say thank you and then introduce Dr. Nelson, and we'll reserve questions for after that. So I will stop sharing. Okay. Brad, do you want to turn on your camera? Okay.
Here I am.
Great. I have an intro, but I lost it. So I'll say Brad Nelson is a fantastic collaborator from the BC Cancer Agency. He leads the clinical development of CAR therapies, the CFIL. Maybe Brad, because I'm butchering it, I'll let you introduce all your titles, and I'll just say Brad has been a fantastic collaborator and real key to the success of the program. The CAGT program has been a collaboration with Brad, so I'll pass it to you.
Thanks, Scott. Yeah, so I'm based at BC Cancer, as mentioned, in Victoria, and I have appointments at UVic and UBC as well. And I'll be presenting on behalf of our CAR T team. So I'm going to pick up on where Scott left off and talk about applying CAR T cell therapy to solid tumors. So first of all, my disclosure is I'm going to talk today about what I think is a very exciting technology that was developed at BC Cancer and UBC and UVic, a collaborative effort that led to a startup company, Novakind Therapeutics, that I have these involvements with. That'll be toward the end of the talk. And so just to set the stage, what is it about solid tumors that presents a challenge? Well, it's really about the tumor microenvironment. And for those of you in the East, you've already had lunch, so I hopefully haven't spoiled your lunch. For those of us in the West, I hope I don't. But I kind of like this analogy because I think there's a good biological basis to it, which is when we think of blood cancers, especially like lymphomas, myelomas, those are relatively tender tissues that are relatively easy for CAR T cells to infiltrate and attack. When we talk about solid tumors, it's like other cuts of meat that are can be really tough, sinewy, gristly, difficult for the T cells to enter. And then when they do enter them, they can stall out or get inhibited, or a variety of forces start opposing their activity. And so far, there's been only anecdotal successes really with CAR T cell therapy for treating solid tumors, such as those of lung cancer, breast, prostate, colorectal, ovarian, pancreatic, and many others. So we want to take on this challenge, and we hope to prevail. And so we've teamed up with the NRC to really build a better CAR T cell to treat solid tumors. And the target antigen that we have rallied around is called mesothelin. And this is just an image from ovarian cancer showing mesothelin expression in brown. So mesothelin is on the cell surface of tumor cells. This is a nice example of really good staining in a tumor. It's found on ovarian, endometrial, pancreatic, and many other cancers, but we'll be focusing on those three. And so starting a number of years ago, we collaborated with the NRC, who have great nanobody against mesothelin, using the same platform Scott's just talked about. So Julian Swazinski has led this effort for our group at BC Cancer, and it's been a close collaboration with Scott and Rossini. So I'll skip all the antibody development. That was done at the NRC, and they did a fantastic job. They sent us several leads. We plugged them into the CAR backbone that we're using for the CLIC-01 and CLIC-02 trials. And this, again, is work led by Julian in our team, and just showing some results from animal studies. So this is an ovarian cancer model, the OVCAR3 tumor line. That's a human ovarian cancer cell line implanted in immune deficient mice, NSG mice. And what we're showing here is tumor sizes of animals post tumor implant. And each line here is a different mouse. And so you can see this is an aggressive tumor, grows very quickly in untreated mice. A benchmark that we used is the SS1 more conventional CAR design and SCFE design that's been used by people like Carl June, Steve Rosenberg, and others in the clinic. Unfortunately, I commend them for doing early trials in this space, but they did not see great clinical efficacy, and nor do we particularly see good efficacy in mice. But with one nanobody in particular of the four that we tested, one of them from the NRC turned out to just be really fantastic in terms of we reproducibly, reliably get complete durable regressions of established tumors in mice. And you can see here 100% complete response rate. And these are long-lived, and we can even re-challenge the mice. I'm just going to get my laser pointer going here, and I'm going to move people's faces. We can re-challenge the mice and see rejection. So there's even a memory response that forms. Just a little more data looking at this nanobody. So again, comparing to the benchmark SS1 SCFE-based CAR, this nanobody-based version, this is looking at CAR T cell numbers in blood over time after adoptive transfer. And we can see we get really nice expansion and persistence of CAR T cells. And those are both really important factors for getting good efficacy. And this is what this looks like actually looking in tumors, just to give a visual impression. So these are multiplex IF images from mice that were treated with CAR T cells. And so this is just showing the mesothelin expression in this teal color in the tumors. So again, you can see it's on the cell surface. Here are the CAR T cells, both the CD4 and CD8 subsets infiltrating the tumor. Cleaved caspase 3 is revealing the apoptotic tumor cells, so we can see those tumor cells just being blown up by the CAR T cells very effectively. And then in orange, we've got the macrophages coming in, the garbage collectors of the immune system coming in to clean up the debris. So this is a very powerful response that this CAR is elicited. And so building on or on the basis of these results and others that we've obtained, we're going forward now with a phase 1 clinical trial of this nanobody-based mesothelin CAR T cell, and we're calling this trial CLIC-04 in our CLIC series. This is the series that Scott introduced, and our clinical leads are Drs. Hannah Chernowski and Yvette Drew at BC Cancer and Vancouver General Hospital. Much like our other CLIC trials, the CAR T cells will be produced in Victoria, and the clinical sites that we have lined up for this are in Vancouver, Calgary, and Ottawa. Plasmid manufacturing will be done by Rob Holt's group in Vancouver, and lentiviral manufacturing will be done, sorry, I'm pointing at the wrong thing, at OHRI, the Biotherapeutics Manufacturing Center at OHRI. So we're excited about this trial, and we, of course, are going into it hoping for the very best for our patients. We can also anticipate, though, that just looking at other solid tumor trials, that there are going to be issues encountered. And so the things that we can anticipate is, in some patients at least, we'll probably see suboptimal expansion and persistence of the CAR T cells. We may see suboptimal infiltration of the tumor microenvironment. Even if things go perfectly well in a patient, there's the risk of selecting for antigen loss variants because mesothelin is rarely expressed on 100% of the tumor cells. And then we're also concerned about off-tumor toxicity against healthy tissues that express mesothelin. So again, this applies to really any T cell therapy for solid tumors, and this trial will be no different. So this is where I'll start talking about our Innovakine technology and how that can potentially help. So just a little bit of immunology 101 here, but for full T cell activation and expansion, you actually require 3 signals. And signal 1 and signal 2 are antigen and co-stimulatory signals. And those are what CARs are good at generating. That's what they are designed to generate. But to get a full-blown T cell response, you also need cytokines involved. And the classic cytokine is IL-2, others are IL-7 and IL-15. And these give you an extra wave of T cell expansion, and they also turn on all the tumor killing genes in T cells or ramp up their activity. So again, CAR T cells give you signal 1 and signal 2. There's a number of ways to get signal 3. It's actually one of the reasons patients are lymphodepleted prior to CAR T therapy. I won't go through all of these, but they all have the same problem, which is they're critical for efficacy, but they're also the greatest source of toxicity. So with our Innovakine technology, we've developed a chimeric GCSF IL-2 receptor, which we call G2R. And this allows us to really precisely deliver signal 3 to T cell products. And I'll just walk you through this. It's actually a very simple idea. It's a 2-part receptor. The extracellular domain binds GCSF, and the intracellular domain in our base model generates an IL-2-like signal in T cells. So why did we choose GCSF? This is also known as Neupogen or Neulasta. It's FDA, Health Canada approved. It's been given to over 1 million cancer patients. It's used to boost white blood cell counts after chemotherapy, as well as CAR T, TCR-T, and TIL therapy. So it's widely used to boost neutrophil counts, very safe. And so we've repurposed it with this receptor. So we can now use the same drug or biologic to induce a proliferative and tumor killing signal in T cells. So when we express this receptor G2R in T cells, this is some Western blots just showing that we very closely emulate an IL-2 signal, but now in response to GCSF. I'm showing you here as well that we can now grow the T cells on GCSF just as nicely as we can grow them on IL-2. So it works as a signaling molecule. And when we talk to T cells about this, the thing we most often hear back is, I can't believe it's not IL-2. They are really fooled into thinking that's what they are seeing. So what about in vivo? So this is a melanoma model, not a CAR T model, but nonetheless a very aggressive mouse melanoma model called B16F10. Many of you in the audience might work with this. And so here we've taken some wild type mice. I'll emphasize there's no lymphodepletion here, just wild type mice. We implant the melanoma, and then we give them T cells that recognize the melanoma, P-mel T cells, that are transduced to express a murine version of G2R. So in go the T cells, and then we randomize the mice to receive either GCSF, and we used a pegylated version in this experiment, or IL-2 as a control, or vehicle as a negative control. And our goal was to determine whether these engineered T cells could eradicate melanoma without the need for lymphodepletion or IL-2. And I should mention this is work done by Sam Preshaw and Megan Fuller. So first of all, just looking at the blood counts in these mice, we see just really whopping T cell responses in peripheral blood. So pay attention to the blue line here. That's the T cells that have our receptor and were treated with GCSF. Note this is a logarithmic scale as well. So well over 100-fold increase in T cell proliferation in response to GCSF. All the other groups are down here sort of in the weeds, including the IL-2 group. You might wonder, why didn't IL-2 work better? It's not because we were trying to make it look bad. We gave high dose IL-2. In fact, we were just going for equivalence in this experiment. But this kind of just shows how poor IL-2 is really at enhancing T cell responses. Also note that the cytokine dosing was for the first 3 weeks, but even when we stopped GCSF treatment, we got a nice persistence of the cells. When we look in tumors, this is day 4 after adoptive transfer. You can see that with GCSF treatment, we get much stronger infiltration of the tumors by T cells. This is CD3 staining for T cells. And then in terms of tumor regression, again, the blue group is the one to pay attention to here. In this study, we saw 4 to 5 of the mice undergo a complete and durable regression of their tumors. Again, there's no lymphodepletion, there's no other treatment. It's just T cells plus GCSF. And you can see the other groups for comparison. And these are durable responses. We've had mice live well over 100 days without relapse. So overall, across multiple experiments, we're getting about an 85 to 90% durable complete response rate. So not shown, yes, we do see neutrophil expansion in these animals, and we can control that largely with the dose and schedule we give. There's no observed toxicities. We can re-challenge and get rejection of tumors. And we've done experiments too where we actually can repeat the GCSF dosing to get a second wave of T cells when needed. So back to the issues and how this can potentially solve them. So we now have means for on-demand, clinician-controlled boosting of T cell proliferation. We can do repeat dosing if we need to. We get safe targeted delivery of potent tumor-killing signals. And we have next-gen designs that go beyond IL-2 and generate other signals such as IL-2 plus IL-12. In terms of antigen loss variants, this technology doesn't address that directly, but it does allow you to not lymphodeplete. And so that means the host immune response is still there. And it also means you can now think about combining this with cancer vaccines or other forms of immunotherapy, again, because the host immune system is left intact. And so in terms of off-tumor toxicity, again, it doesn't directly address it, but does give you a safety mechanism. So essentially a throttle that you can either throttle up or throttle back. So if there's toxicities, lower the dose or discontinue GCSF. So in conclusion then, engineered cell therapies are having impressive success against blood cancers. Scott nicely showed that. Solid tumor barrier is the next frontier that we all need to tackle. The NRC and BC Cancer's nanobody, Mesocar, rocks as far as we're concerned in mice so far, and we're excited to move that to the clinic. There's ways to, as I've shown you with the Innovakine tech, and there's other ideas too, to improve on features of T cells. And importantly, this work is all being done in Canada for Canadians. And I think this is exactly what we need to be doing in Canadian research at this time. So I'll stop there.
Thanks, Brad. Fantastic. So I will quickly introduce Dr. Lee, but we will have 5 minutes for questions after this session. So I just want to, if you have questions, go ahead and start putting them in the Q&A. I think Teffer's going to collect those, and we will have time at the end as well. So just to let everyone know, we'll have hopefully adequate time for lots of questions. So Dr. Lee is a full professor in the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa, where he's held a faculty appointment since 2011. He completed his Ph.D. under Dr. Sylvia Vidal at the University of Ottawa, and working on host cytomegalovirus interactions, and led to the identification of host resistance genes against CMV. He subsequently pursued postdoctoral training in the laboratory of Dr. Christine Biron at Brown University, where he characterized key cytokine pathways governing NK cell function. And now, with over 25 years of investigation in NK cell immunoregulatory biology, Dr. Lee's current research program is focused on translating foundational knowledge of NK cells into immunotherapy. And this has been really a fantastic collaboration. I'm really excited. Dr. Lee, please.
Okay, so Dr. McComb, thanks so much for your kind introduction. Let me share my slide first. Can you see my slide well?
Yeah, looks great.
Okay, so good afternoon, everyone. My name is Seung-Hwan at University of Ottawa. So before begin, I'd like to thank BioCanada for the invitation to present today. So I'm very excited to share our collaborative work with the National Research Council of Canada on NK cell-based immunotherapy against multiple myeloma. So let me start the biology of multiple myeloma. Multiple myeloma is the malignancy of plasma cell in bone marrow. In healthy individual, the plasma cell, the terminally differentiating B cell, producing the protective antibody, actually protecting us from many different infections. However, in myeloma patients, these cells become cancerous, just the proliferating in controlled manner, and then secreting this the useless antibody. So what makes the myeloma particularly challenging is immune evasion. So the multiple myeloma cell is not intrinsically recognized very well by our two major cytotoxic lymphocyte population, T cell and NK cell. So this means we need an artificial receptor to redirect immune system to eliminate the cancer cell. So one of a very well-known example of artificial recognition system is the CAR, chimeric antigen receptor. The two CAR T cell products are now FDA approved for the myeloma, and then this cell, and then the CereCura cell. The both the targeting BCMA, the B cell maturation antigen, which is highly expressed in the myeloma cell and then also the late stage of plasma cell. So making it an ideal target. The therapies work very well with a very impressive the clinical efficacy, but they comes with a serious limitation. They induce significant side effects. But the real problem is that those CAR T cell therapies are given in autologous setting. So we must collect T cell from the patient, and then engineer and expand them, and then infuse them back to the patient. So this process is logistically demanding and extremely expensive, leading to this high price tag up to $500,000 per person. So if we try to use this the CAR T cell in allogeneic setting, we face, to those, the life-threatening, the graft versus, host disease. So taken together, these, limitation define and clear unmet need, encouraging toward an allogeneic off-the-shelf therapy with a better safety profiling. So NK cells are compelling answer to allogeneic therapy. Unlike T cell, NK cells do not cause, the GvHD, and also they exhibit very, very mild, the side effect, based on the clinical experience, up to date, but still holding a very strong anti-tumor activity, and then demonstrating their potential, like the very strong potential for the off-the-shelf therapy. So to improve, those allogeneic NK cell therapy, currently we try to harness, the ADCC, antibody-dependent cellular cytotoxicity, using the antibody or engager, and also we apply the gene editing using CRISPR-Cas9, and also we express, the CAR on NK cell, which is the main topic of my talk today. So landmark clinical work, from Dr. Rezvani at the MD Anderson Cancer Center have validated CAR-NK cell approach. So especially this, their phase 1 and 2 clinical trial, the data published in Nature Medicine in 2024, they use, they target, B-cell tumor using allogeneic CD19 targeted CAR-NK cell. So the result is really striking or dramatic. A 1-year overall survival of, 68% and 94% of, the response rate. So all of this, without severe side effect or GvHD. So this data really strongly established NK cell-based, the therapy as a next generation immune cell therapy. So in NK cell immunotherapy, CD19 or NKG2D ligand are the, popular target, and then also BCMA is one of, top. And then disease-wise, the NK cell trial is concentrate on the hematological malignancy, but it's gradually expanding to the solid tumor, and then more recently, autoimmune disease. So as you can see, those disease, mostly like hematological malignancy, but you can see the many like a solid tumor, and then the lupus or autoimmune disease now is also being explored. Geographically, China actually lead with almost 300 active clinical trial, followed by the U.S. They have roughly 100. But what I want to draw our attention, your attention, that the Nielsen mentioned, there is actually we have a really more Dr. McComb and Dr. Nielsen mentioned, like we have a really like the need for the cell therapy. So there is currently no active NK cell clinical trial registered in Canada. So that gap is exactly the motivation our program. So to address our gap, we established a structured collaboration with the NRC, the CGT, the program with 2 CGT projects, the 501 and then 807. And then so our program has 4 integrated aims. The first aim is the designing and optimizing CAR construct targeting multiple myeloma. And the second thing is developing bispecific and trispecific the engager with NK cell against the multiple myeloma. And then third is optimizing lentiviral transduction for NK cell engineering. And then more recently, we optimize and establish GMP compatible manufacturing pipeline for clinical translation. So I'm very pleased to share some of what we have accomplished so far. So cell manufacturing process development to generate many number of NK cells with a potent anti-tumor activity is a critical component in NK cell therapy. So we established GMP compliant CAR-NK manufacturing pipeline in collaborating with NRC and Bakcell Bio in South Korea. So Bakcell Bio is actually providing the feeder cell system, which is a critical component for NK cell expansion. So the manufacturing pipeline is built using the G-Rex, the platform. So those G-Rex, technology is straightforward but also powerful. So A gas permeable membrane at the base enable efficient oxygen nutrient the delivery by convection, the supporting very high cell density with a minimum the media change. So for example, so this is more than the 50 of T75 flask can be replaced by the A single G-Rex plate. And moreover, this system is scalable and then GMP compatible and dramatically simplify workflow. So in practice, using G-Rex centric expansion protocol, we starting from the 120 ml of peripheral blood or cord blood, we isolate NK cell, and then we expanded NK cell using Bakcell Bio feeder cell system. And then we could generate, this is a hypothetical number, but we could generate the 2,000 billion of expanded NK cell. So when NK cell is just freshly isolated, they are very small, but when they are stimulated with the feeder cell, they are actually enlarged. And then this, the large NK cell are highly cytotoxic. As you can see, their folds and the side scatter, the data also show they are highly proliferative characteristic. So this pipeline is really reproducible, scalable, and then clinically applicable. So to demonstrate that our expanded NK cell is actually functional, we test them with the bispecific, the killer antigen. This is the NRC generate the bike. And then this bike actually the link the NK cell and target cell, because this bike can bind NKP30 on NK cell and then BCMA on target cell. So in the NHG genogram model using luciferase expressing MM1 cell, the combination of IL-15 expressing NK cell plus those the BCMA bike dramatically suppress the tumor burden. As you can see, NK cell alone, it doesn't really show dramatic effect. So that is really demonstrate the superior cytotoxicity of NK cell from our manufacturing pipeline. So let me turn to our central story, CAR-NK therapy against multiple myeloma. So this project was led by my Ph.D. student, Shelby Kaczmarek, and then Elisa Bisram, Jurabinska, Cancer Center in McMaster, and then Scott McComb at NRC. So NRC contributed a critical piece to our collaboration by providing their established, the expertise in generating the high specific nanobody. So here, like the llama immunization using the BCMA exodomain is producing a single domain antibody, which is really the highly specific and then highly, the high affinity. And then those, the binder now is incorporate into the CAR construct. And then we receive those, the construct and then express, in the NK cell, and then we test their anti-tumor activity in vitro and in vivo. So the first, CAR-NK testing show, our successful, like the genetic engineering of NK cells, so we can really overexpress NK CAR expression very well. And then also, it shows excellent in vitro activity against the multiple myeloma cell, which is MM1 cell expressing high the BCMA. So this is CAR negative cell, CAR positive cell. It's dramatic, the anti-tumor activity. However, in vivo result was not quite impressive, only delaying the myeloma growth in vivo. Then we realized it was due to the limited persistence of CAR-NK cell in vivo. So 3 weeks later, is very few NK cells remain. So NK cell persistence in vivo is a fundamental bottleneck. Without sustained cytokine signaling, transferred NK cell usually rapidly contract and disappear. So to overcome, we co-express IL-15 with the CAR, and as you can see, CAR-NK cell, this is the CAR-NK cell without IL-15, or this with IL-15. So after 5 days, the culture without IL-2, actually most of the cells is found there, but with the IL-15 co-expression, actually all the cells actually they're very, they still survive. And then the InQ site data actually show this is the without NK cell. This is the CAR-NK cell without IL-15, CAR-NK with IL-15. So it really shows the much more potent anti-tumor activity. The more importantly, they remain well in vivo for more than 1 month. So using this, the CAR-NK cell co-expressing IL-15, we repeat, in vivo experiment, and then our in vivo experiment clearly show the importance of CAR expression, because as you can see, a modified CAR-NK cell does not induce any benefit compared to those untreated group. And even the IL-15 expressing CAR, they still doesn't show any benefit. As again the CAR-NK cell, they delay the myeloma cell growth in vivo, but as you can see, notably, CAR-NK cell co-expressing IL-15 clearly demonstrate effective tumor clearance. So cell analysis at the end point also show CAR persistence is extended with the IL-15 co-expression in blood and spleen and bone marrow. And more importantly, analysis of myeloma cell in bone marrow. So as you can see in the end point, the myeloma in the bone marrow is really full of myeloma cell. But the CAR-NK cell is co-expressing IL-15. They show really the dramatic, the effective, tumor clearance. And then as you can see, almost no myeloma cell is found here. So to summarize our finding, the CAR-NK cell expressing IL-15 demonstrate, the potent anti-myeloma activity in preclinical xenograft model, and then with the markedly extended in vivo persistence, and then superior tumor elimination. And then we also have established a GMP-compatible manufacturing pipeline ready for the clinical trial. So looking ahead, our priority, our future plan, is scaling the G-Rex expansion and then lentiviral the transaction to clinical trial volume, and then also we try to combining our CAR-NK platform with a complementary modality such as the bike or multi-targeting the CAR construct, and then also we want to completing the safety and the toxicology and regulatory the package required for clinical trial application. So our ultimate goal is to bring a safe, effective, and then truly accessible off-the-shelf NK cell therapy to cancer patients in Canada. So finally, I want to thank our lab member and then our collaborator in NRC, and then also Dr. Elisa Bisram and Jurabinsky, the Cancer Center, and then our partner at Baxel Bio. And also I'm grateful to all our funding agencies for their support. Thanks so much for your attention.
Thanks so much, Seung-Hwan. Excellent. So while we're bringing up the questions, maybe Brad, I know you didn't get a chance to show your acknowledgement slides, so please do that, and maybe I will quickly, go ahead, please.
Okay. Let me just see. Is that showing?
Yep.
Yep. Okay. Yeah, I got so excited talking about CAR T cells and et cetera that I forgot my very last slide, which is, of course, the most important. So I just wanted to highlight and thank my team in Victoria, the folks in Vancouver involved in this, clinicians, the NRC, of course, have been great partners in this. And then we're doing the biomanufacturing of the lentivirus at the DMC. And our patient partner, Aaron Barrett, has really been foundational in this, and of course our funding. So NRC not only gave a great binder, but has gave us funding for this project, as well as BioCanRx, another sponsor of today's event. So thanks to all involved.
Right, so I think we'll take just 5 minutes of questions, but I will say we've all of the speakers have said they can go past the hour. So if there's really burning questions, we will stay on after 3 o'clock. So do stick around if you want some more time for questions. But Teffer, is there something in the Q&A we should?
There's no one in the Q&A
Okay.
yet, so hurry over.
Okay. Yeah, okay, well, that's great. I mean, if anybody wants to raise their hand, we could. I think there's no questions, then we'll just jump to the next session and we'll save questions for the end. I'm sure we can have some discussion. I can talk about this subject, as everybody probably knows, forever. So, but I think in the interest of time, if we have no questions, then we will pass it to Umar and get on to the subject of non-viral gene therapies, which is also very exciting and will be, I think, of great interest to the audience. So, Umar, do you want to? There we go. All yours.
Thank you. So let me just put this in full screen. So yes, I'm very grateful to be able to speak here, and I'm excited also to be followed by 2 very good collaborators, Dr. Maganti and Dr. Weilinger, which I'll introduce after. But just to kind of open up the area, I'll be talking about tissue-targeted delivery of lipid nanoparticle-based therapeutics. This is like work supported heavily by the NRC Cell and Gene Therapy Program and also other funding sources, which I'll talk about later. I think the key here is really to understand that lipid nanoparticles, or LNPs, they're clinically validated. They tend to be cargo agnostic delivery systems for nucleic acids. However, the main challenge with them has often been that after systemic administration, you have a lot of accumulation in the liver. Now, today I'm going to discuss kind of NRC's approach to try to overcome this challenge, which involves kind of 2 tiers, trying to improve on the lipid nanoparticle formulation for more organ-tropic kind of biodistribution, but also involving cell-specific targeting peptides or ligands or antibodies. And I think together with these approaches, I mean, I think we're moving toward a more programmable platform that can deliver different cargoes to extrahepatic organs and novel cell types. And I'll go through a couple of case studies in different organs, including the brain, skeletal muscle, T cells, and Schwann cells. Now, just to give you kind of, for those who don't know, but LNPs usually consist of 4 lipid components. You have what is known as the ionizable cationic lipid. This is the one that helps encapsulate the nucleic acid and enables endosomal escape when you're inside the cell. You also have kind of a helper lipid that supports the structure, and cholesterol, which also gives a lot of stability in vivo. And then finally, a PEG lipid, which helps keep it control the size of the LNP. So it's it is suitable for the body, and also it helps in circulation time. And you can attach ligands to any of these components, but in particular to the PEG lipid. Now, each of these lipids are tunable, and by tuning them, you can really alter the potency of the delivery, the stability, and the distribution of the LNP. And we have already seen very good clinical success, like by Onpattro, which is done in 2018 approval, and COVID-19 vaccines in 2020, 2021. Now, this slide is really to indicate that LNPs are highly versatile. It's a highly versatile platform. It can deliver a spectrum of nucleic acid payloads. As you can see here, like depending on the disease, you may need a different genetic intervention. So, for example, one could do gene addition, so replace missing proteins using, say, mRNA or some type of DNA. You can have gene silencing, so maybe you want to reduce harmful gene expression with antisense oligonucleotides or siRNA, and gene editing, again using CRISPR technology, and in going further into base and prime editing, and then gene regulation, which is a bit newer, but it allows you to regulate the gene without editing. So maybe you want to turn on a gene or off a gene. Maybe you want something short-term or longer term. Longer term allowing, say, for by using, say, epigenetic editors. But the key here is that the biology determines the therapeutic strategy, and the modular nature of the LNP allows you to deliver that technology. It's important to understand why the LNPs can go to the liver. It's not an accident. It's actually part of the biological programming. So when something enters the bloodstream, serum proteins, in particular ApoE, can coat the LNP surface, and ApoE has a receptor like LDL receptor, which is present on hepatocytes. So these LNPs are then actually actively internalized in liver cells. And for a liver disease, this is great. It's an advantage. And for non-liver diseases, it's a challenge. So the challenge being that only maybe a fraction of the dose reaches other target tissues, and causes you to go higher dosing, increasing cost, or maybe increasing toxicity. So the goal of the bioengineering goal really is how to overcome ApoE-driven liver uptake and actually redirect LNPs to specific extrahepatic sites. So the way we approach it is actually like more of a two-tier strategy. And that often works synergistically together. So tier one is really trying to improve on organ tropism or organ targeting. And this is mainly achieved through the lipid formulation. So us and many others, they will modify the lipids, adjust maybe even the surface charge, or add known lipids that are considered SORT lipids, so selective organ targeting lipids. But the goal here, the purpose of that, is to try to get particles away from the liver and into other organs. So this has already been quite successful for some organs like lung and spleen, and it's slowly and quickly, moving toward other organs. And these surface changes also affect how serum proteins can coat and which serum proteins will coat. And in other cases, the lipids have a strong preference for some organ type, so they themselves are driving organ tropism. So let's say we're getting to the organ of interest. Now we have to get to the tier two, which is getting to the cell of interest. So this is why cell targeting is very important. And we generally achieve this through like adding surface ligands, such as antibodies or peptides or other targeting moieties onto the surface of the LNP. And then these are carefully selected so that they're unique to, say, a certain cell type, but also that they can trigger a fast internalization, because you want the LNP to be bound, but not to stay bound on the outside, but to be driven inside. So internalization is also very important. And then once internalized, the LNP has a natural ability to escape and deliver its contents into the cytosol. But the purpose here is really to get particles into the correct cell type. And generally, both matter. I think we can't have one and that's sufficient, or the other, but we need both because organ targeting, will lack cellular precision, and ligands alone may not overcome, say, the liver accumulation. So together, you have the potential of both improved tissue and cell-specific delivery. Now, for this, just some examples of tunable lipid formulations that help drive organ tropism. We often use like reporter mRNA, such as those that encode firefly luciferase. It allows us to image using imagers that detect what is known as bioluminescence. And this mRNA is easily encapsulated into LNPs, and these LNPs will have different lipid compositions. So on the left, for example, we are trying to show here that, okay, we can have an LNP that is more of a longer circulating LNP. It can reach many, many organs beyond the liver. Again, some diseases may want a more widespread delivery. So, these could be useful there. Also, by having longer circulating LNPs, you have more chance of, say, targeting them to different, more of the dose can be targeted, say, to different cell types. In the middle, again, by changing the lipid composition, we can drastically affect the biodistribution and have, say, more organ tropism to, say, organs like the spleen. Spleen is an organ that may where many immune cells reside. So bringing LNPs there is one step, but then targeting the cell type of interest, again, is a second tier, which again would usually need a targeting ligand. And then on the right, again, we are working towards trying to get more organ tropism to the blood-brain barrier and to the brain. And this is an ongoing work, and a very exciting area as well. So as I was explaining before, yes, reaching the organ of interest is definitely the first step. But then once we're there, we need to get to a specific cell within that region. And generally, we like to do this with antibodies. As you can see here, we have the conventional antibody, but these are a little bit big, so we prefer to use fragments. One particular antibody we like is actually the single domain antibody, which is really the smallest antigen binding domain that exists in nature. So it's easy to, it has high affinity and it's very stable and can be humanized, and it can be engineered to have site-specific handles that we can link to the LNP surface. So that allows us for keeping the orientation and controlling the density of the targeting antibody on the LNP. And NRC has a kind of a large and growing portfolio of antibodies for various applications. In other cases, we also use other targeting moieties where we have worked with peptides. Some of them are very high affinity and specific for receptors. And then we're also exploring other technologies like aptamers and seeing, how well they can target. But at the same time, there's the benefit of being very stable and very low cost. Now I'm going to show you a number of case studies. One being brain delivery using what is called a blood-brain barrier transmigrating single domain antibody. So as you know, there is a large challenge to cross the blood-brain barrier, for all biologics, whether antibodies or viruses or cells, and including LNPs. You have to cross before you can actually reach the, say a neuron. So the blood-brain barrier transmigrating single domain antibodies, we have a large panel that we've worked on over the last couple decades. The idea behind them is that they bind receptors that are selectively enriched on brain endothelial cells. For example, the classical example would be transferrin receptor. And after binding, they undergo what is known as receptor-mediated transcytosis. So to actually not just be taken up, but to actually transcytose or cross and bring the cargo, like the LNP, across into the brain parenchyma. So here on the right, middle and right, we are, working on optimizing the delivery of BBB targeted LNPs after IV injection, and our goal is really to get therapeutically relevant concentrations of the cargo into the brain, which is very, has always has been a challenge. But by using LNPs, our hope is that we can, have kind of open up this field and where others have failed to actually get some of this newer technology into the brain. So, and another example, another targeting example, that we're working on is actually targeting skeletal muscle. There is quite a bit of muscle in the body, but to target that, you need something very specific to that cell type. So we're using single domain antibodies as well that are muscle targeted, linked to the LNPs. And we're trying to, with our collaborators at Sick Kids, trying to test this in a model of myotubular myopathy, which is basically a loss of the MTM1. And this loss of MTM1 it leads to very severe kind of weakness and respiratory failure, like especially in children. So the goal here is to say, can we deliver, reintroduce the MTM1 by delivering MTM1 mRNA and actually restore the protein expression, and without having anything integrating, maybe a redosable and a dose adjustable manner, because the LNP has that flexibility. Just showing you some preliminary data, like say if we look at the middle panel, we are treating mice that are very severe case of MTM1 deficiency, and we're giving these LNPs to deliver the MTM1 mRNA, and we're showing that after some repeated injections, we're able to kind of have the mice have an increased hang time, which is a measure of their muscle strength, and also on the right side, increased velocity. The mice are very crippled, but then obviously if you start to correct or deliver the MTM1 mRNA and then the resultant protein, you can have some recovery of the muscle. So it's a ongoing, but this is like at least showing positive that the targeting and the delivery can be done via LNPs. And related to some of the previous talks, T cells are a big focus in the field, and many immune cells. But we are working also at maybe delivering CARs in vivo. So this is really in vivo cell engineering or T cell engineering. And with some collaborators, Dr. Megan Levings at UBC and SPME, we are trying to see if we can achieve this by using CD3 targeted LNPs. Right now, as we explained earlier, the ex vivo kind of CAR T manufacturing is very has challenges. You have collect cells, you need viruses to transduce, and there's also the expansion and the reinfusion. So there's a cost and time to that. But if you can theoretically, or if you could give targeted LNPs that are hitting the T cells in situ, like in the body, and these circulating T cells or those in the spleen, you can deliver, say a CAR mRNA. In this case, we're looking at delivering, some bicistronic mRNA, like with including FOXP3, which can help change the T cell identity to be more Treg, and also HLA-A2 CAR, which will help give antigen specificity. These could transform some of your body's T cells and make them targeted so that they are useful in indications like autoimmune disease or tolerating transplants. And the key here really is just, can we replace some of this ex vivo therapies with more off-the-shelf IV therapy, giving something a bit simpler and giving patients better access. And the last example I wanted to talk about was just, like there are, yes, we talked about a bit immune cells, but there's also many cell types, and diseases. So here's, more, not like, a disease that is very specific for Schwann cells. It's called CMT1A or Charcot-Marie tooth type 1A. And this is not a disease of a lack of a protein, but actually too much overexpression of a PMP22 gene. So the goal here is actually not to replace but actually to dial back the expression of this gene. And one way to do that is actually to deliver silencing molecules like siRNA, or maybe even working at the epigenetic level and trying to silence the translation of the gene. So here, just to show, in this case, we are looking at peptides that are very specific for Schwann cells and not other types of cells, and demonstrating here on the right that, let's say in the top panel, we are selecting LNPs that have, better delivery naturally, baseline delivery. And then number two, below that, we are looking at targeted LNPs that are Schwann cell targeted LNPs. And we're showing that we can deliver the contents, much more efficiently, which is, very good. And this, again, to working toward, getting, this is a gene that you don't want to, you have to be very careful not to over silence. So a lot of tailoring and the dosage will be very important, especially if one were to go to animal studies. And my last slide here. I just say, the ultimate goal really is this programmable medicine. So, using LNPs with different cargos and with precision targeting. We know LNPs are modular by their design, and they can encapsulate many different nucleic acid cargos. And as I explained earlier, the targeting, really should be two-tier. So yes, maybe there's an organ tropic formulation, or combined with surface ligand specific targeting. And there's a growing list of organs and cells being targeted in the field, like in the literature. And really, we want to get to pharmacologically relevant, in vivo cell engineering that is like non-viral and redosable, and again, hitting the new cells and organs that we wish to. And finally, yes, I mean, this is not a comprehensive list, but there are many, many people to thank. I just had a small list here, and a lot of support from NRC and other organizations for, in terms of working in this space. Okay, so I think we're going to continue. So it's my pleasure to actually introduce Dr. Harinad Maganti. He's a research scientist and a scientific director of cellular therapies at the Canadian Blood Services. His research is focused on developing innovative technologies that accelerate the translation of cell and gene therapy products from laboratory to clinical application. And him and I, we share a vision of trying to get more items, just like, more products that are made in Canada. So I think I'll pass it off to him to introduce his talk. And yeah.
Thanks, Umar. Just gonna, okay, great. Thank you. Could just someone confirm that they could see my slides and not my notes, but my slides? Yeah.
Yes.
Yeah. Very good. Thank you.
Okay, awesome. I want to thank you again for this opportunity. Today, I'm going to talk about some of the really neat work we've been able to do in collaboration with the NRC. Specifically, I'm going to focus on stem cell gene editing. So this work started a few years ago. It was specifically funded through the NRC ideation, as well as the cell and gene therapy grants, which subsequently led us to go and get external funding from the American Society of Hematology, as well as funding from Canadian Blood Services. And this is a great example of how personally my team has really been able to use the cell and gene therapy program, leverage the funding to get more extra funds. I don't have any conflicts of interest to disclose. In my talk today, I'll be using the word we, but the people that have, at least on the front facing, that have done a lot of the work are these three people. So Justina Anoto is a Ph.D. candidate in my lab. She's done a lot of the the gene editing work that I'm going to be talking about in collaboration with Chelsea McGregor, who's a very talented lab manager and research assistant. And from the NRC side, we've been working a lot with Umar as well as Dr. Brian Samard, who's really helped us make the actual lipid nanoparticles. So you've heard a lot about, cell and gene therapy products from the immunotherapy side, but let's talk now about stem cells. So stem cells are the mother of all blood stem cells. I'm a stem cell biologist, so I'm a bit biased, and I think they're very important. But stem cell gene editing can lead to several curative therapies. But however, the two major issues that the field is facing with HSC gene editing is maintaining the stemness of the HSCs ex vivo. So how do we target them? And the second is actually delivering gene edited products into these cells without inducing cell death. The two most common methods by which gene editing machinery is delivered into stem cells are electroporation, which often leads to up to 50% cell death. And the second method is through lentiviral transduction, which has its own issues, with random integration being one of them. So sickle cell disease is a model. This is the model we're going to use and talk about today. It is caused due to a point mutation in the beta globin gene, which makes the red blood cells go from their this perfect circular state to a sickled state. And when they become sickled, they have trouble moving through the blood vessels, and it leads to clumping and causes a lot of pain episodes for patients, unfortunately. The other issue is that they're not able to carry enough oxygen, so it leads to a lot of issues in terms of organ damage and stuff. So many patients end up being diabetic, for example, and it's really unfortunate. There are over 8 million people in the world affected by sickle cell disease, and it's highly prone in African American people, 1 in 300. The cases of sickle cell disease are consistently rising in Canada through immigrations. And the cost of treatment, a cell and gene therapy product right now is about $2.5 million per patient. So as you can imagine, it has a very high impact on the health system. And it's provided through Vertex, and the way they go about is they electroporate cells. The guide actually ends up mutating the enhancer region of BCL11A, which then leads to production of gamma globin. So in the bottom here, you've got an adult hemoglobin. The way it has is it's got 2 alpha chains and 2 beta. In patients with sickle cell disease, there's a mutation in the beta chain, so therefore this molecule does not carry oxygen efficiently. The idea is that if we de-repress gamma globin, the gamma will combine with alpha and form a field globin, which then is able to overcome this oxygen carrying capacity problem, as well as it de-sickles the cell into healthy round cells. Now, as I said, the problem with gene editing historically is targeting the stem cells. And so what a lot of these gene editing products happen is patients are often undergo more than one round of mobilization. So they're given a drug, the drug mobilizes cells into the peripheral blood. They go over 2 rounds, and this has this, it's not a comfortable process, but also it increases the production time of the product by up to 6 months, which again feeds into the cost of the treatment. So one of the things we were thinking about is, are there ways we can expand adult stem cells? And now this work has been done previously in the cord product, in the cord stem cell scenario, where SR1, UM171, which is a Canadian discovery, and valproic acid have all been shown to promote cord blood stem cell expansion. However, these molecules are not great at expanding adult stem cells. And what's important to note is that all these molecules work through different pathways. So one of the questions we asked is, well, is it possible that we could combine these molecules using mathematical modeling and machine learning to come up with cocktails that are capable of expanding adult stem cells and meeting their demands? And part of the reason why we went through this path is because we've had earlier success in this, where my colleague, Dr. Nicolas Pineau, his team created stem cell agonist cocktails for expanding cord blood stem cells, and has shown that the strategy works really well. So what we do is first we created these random iterations, 93 different combinations of small molecule cocktails, all of which expand stem cells at different fold. We feed this information into a machine learning program using Bayesian modeling. We create second, third, fourth generation of cocktails. And this work is done in collaboration with Dr. Julie Oddett and University of Toronto. And what this ultimately leads to is it gives rise to some of these cocktails. I've just listed some numbers here. And what you have in panel A is the CD34 count, which is mostly progenitor cells. And as you go to panel C, you're looking at really the enriched hematopoietic stem cell population. And each dot here is a cord blood, it's an independent cord blood unit. So what you can kind of appreciate is that, for example, if we focus on 75 here, this combination expands stem cell population close to 100-fold, whereas the individual molecules are more like 25-fold. And X2A, which is our previous cocktail that's robust at expanding adult cord blood stem cells, is closer to 55-fold. And this is really good and important because in order for HSC-based gene editing to be successful, it's absolutely critical that we have to edit stem cells. And when we talk about editing more complex illnesses other than sickle cell disease, like SCIDX1, wherever, the percentage of efficiency significantly declines. So having more HSCs means patients don't have to go through multiple rounds of gene editing or multiple rounds of mobilization and multiple rounds of gene editing. So now that the first issue has been sort of dealt with, we're going to focus on the second topic. So I'm not going to introduce what lipid nanoparticles are, because Umar's done a fantastic job and is far more qualified to talk about this than me. But the thing that makes us really excited about lipid nanoparticles are that they're a non-viral alternative with reduced risk of integration. They have low immunogenicity and low cost of production. In our case, what we do is we take this LNP, we coat it with a protein or a ligand that binds to a receptor on hematopoietic stem cells. And the idea is then the LNP with the ligand undergoes receptor-mediated endocytosis and ultimately releases the payload. In our case, it's CRISPR-Cas9, mRNA, and guide, which then go translation and ultimately lead to gene editing in the nucleus. So here's the general platform strategy we use. So we take mobilized peripheral blood cells from donors. We expand them for 5 days. We then throw in the LNPs, wait for about 16 to 20 hours, and then we take these cells. We do a whole plethora of experiments where we look at RNA sequencing for transcriptional changes occurring. We do whole genome DNA sequencing to know off-target effects. T7 endonuclease assays for more targeted analyses right away. When we also differentiate these cells over 14 days. And the idea here is we want to know if these gene-edited cells are able to produce gamma globin, fetal globin. Are the genes such as BCL11A and all of those, are they impacted? And also, more importantly, are the gene-edited cells capable of forming red blood cells, which is the most important part of the project. And one of the most important steps is characterizing the LNPs. So we do this in collaboration with Dr. Vera Tang at University of Ottawa using the Flow core facility there. And what we do is we look for LNP concentration, the size of the particle, polydisparity index. And this for us is quite critical because it's important to know the ratio at which LNPs to target cell have to be plated, and because that impacts our gene editing efficiency as well as viability, as well as characterizing the size of the LNP. So one of the first things we did is, and this is largely due to the ease of accessibility, we're at Canadian Blood Services. We have a large cord blood bank for research programs. So we've actually leveraged our cord blood bank, isolated CD34 positive cells, and transfected these cells with different combinations of LNPs and looked for viability as well as transfection. And now these LNPs were loaded with EGFP mRNA. And what we identified is that there were a few combinations that did well, the viability overall was good, and the transfection efficiency was also good. We decided to focus on the LNP4 because it has the It's the most preclinically advanced cationic lipid. So all the subsequent work I'm going to be talking about is going to be with LNP4. Now this work laid the foundation. Now there are, as I've said previously, differences between cord blood CD34 positive cells and adult CD34 positive cells. So we've then switched over from cord blood to adult mobilized peripheral blood CD34 positive cells. Each dot here is a donor. So these results were validated with several donors cells. And what we then did is we replaced the EGFP mRNA with CRISPR-Cas9 guide mRNA, and again, transfected the cells where the LNPs were coated with different ApoE concentration. So ApoE is it's essentially a ligand for the LDL receptor. The idea is that we wanted to know if we increased the coating of the LNP, will it therefore promote better receptor-mediated endocytosis? And we also looked for transfection efficiency, and we identified a sweet spot where we were able to get pretty high transfection efficiency and quite minimal cell death, around 80%. So we went ahead, did the actual experiments where we have a mock control here, which is the same as the LNP, except it doesn't have a guide, so it has the CRISPR-Cas9. The cell viability is pretty good, close to 80%. It doesn't really impact the CD34 percentage viability compared to the untransfected control. And the transfection efficiency was also pretty good. It's over 50%, in some cases over 60%. We did the T7 endonuclease assay where we were able to see that the gene editing was done really well. So in this case, this is the unedited cell. If there was if there was unediting, if there was no editing, you should see some bands here, but this looks pretty good. We quantified this using image analysis, and it showed that we had over 50%. I honestly think there's higher gene editing efficiency, but that's something we're now exploring through whole genome sequencing to study both on-target and off-target gene editing efficiencies. Now, what we also did is we looked at the target genes. So if gene editing works, the BCL11A expression should go down. It's because we're targeting the enhancer region, which is exactly what we see in the LNP compared to the mock. The gamma globin expression also goes up in the LNP compared to the mock, which is quite exciting. And you will see that there are these differences. And this is, again, because of donor variation. This is a biologically relevant thing, and it's not something due to experimental issues. And then, we see decrease in beta globin, and this is, again, has been observed because gamma and beta, there's a crosstalk. And then there's not really big changes in the alpha globin levels. Now, all this is great. This is all transcript, what's happening at the protein level. We did intracellular flow where we looked at the percentage of cells expressing a fetal globin, and you could see that over 85% of cells are expressing fetal globin, which is, again, very robust. We've actually quantified the amount of fetal globin expression using HPLC in collaboration with our partners in Thailand. And what they've noticed is that there's over threefold increase in fetal globin expression compared to the mock and untransfected. And again, this was done over multiple donors, suggesting that this is a quite highly reproducible. And this is the last slide I'm going to show, so won't take too much of your time. What we're looking here is erythroid differentiation. So essentially the way the cells go is they're supposed to go like a horseshoe shape, like this all the way down, and we've quantified these flow plots in panel B here. And what you can appreciate is that as the cells go from erythroblasts to mature erythrocytes to enucleated cells, the percentages don't change, suggesting that gene editing is not necessarily impacting overall the differentiation process of these cells. And we've also looked, here's a Giemsa stain where we're looking at enucleated cells right here, and you could see that there are these beautiful enucleated cells, suggesting, again, the LNP transfection process, along with our expansion platform, works robustly well. So to conclude, what I'd want to highlight is that we have created, I would like to say, a made in Canada gene editing platform that works really well. We've demonstrated its efficacy through sickle cell disease as a model. We're now testing actual sickle cell disease patient samples in collaboration with the clinicians at the Ottawa Hospital. And more importantly, we're doing a whole bunch of next generation sequencing experiments to characterize the quality of this product. But also, this platform that we have is going to be very useful in treating more complex illnesses, such as SCIDX1 or other autoimmune conditions that currently are going to be very hard to treat with existing platforms. I just want to acknowledge Umar and his team. Again, I don't know all the people involved, so I apologize for not including everyone. Julie and her team in Toronto, Alan Tinmouth, the clinician, and his colleagues, as well as my research team and all the different funding bodies, specifically the NRC Cell and Gene Therapy Program.
Thank you. Excellent talk. I think because we're going to do the questions at the end, so I'm going to turn now to our next speaker, Dr. Nick Weilinger. So he's a translational neuroscientist by training with a background in subcellular mechanisms of cell death in neurodegeneration. He completed his Ph.D. in neuroscience at the University of Calgary and postdoc at UBC. And he is currently co-founder and chief scientific officer at CereCura Nanotherapeutics, which is a UBC spin-off company trying to develop the next generation of LNP mRNA therapeutics for patients with brain disorders. And I'll pass it off to Nick.
Thank you, Umar. Can everyone see my slides and hear me?
Yes.
Okay, terrific. So yeah, thanks for the introduction, Umar. Again, I'm Nick Weilinger. I'm Chief Scientific Officer at CereCura, and we are a UBC spin-off developing LNP mRNA-based therapeutics specifically for the brain. We spun out of the labs of Brian McVicker and Peter Cullis here at UBC with really the goal to tackle, with the goal to tackle drug development for neurological disorders as a platform. And so I think it comes as no surprise to anybody that the impact of disorders of the central nervous system is enormous from a socioeconomic standpoint. About 10% of people globally are affected with severe brain disorders. And this is a problem that's only going to increase over the coming decades with an aging population. And so specifically, I'm talking about disorders like injuries like stroke or traumatic brain injury, neurodegeneration like Alzheimer's, Parkinson's, brain cancers, or even monogenic rare diseases like metabolic disorders of the brain. And the cost is enormous. So in the US alone, it's $800 billion a year of both direct and indirect costs. And unfortunately, for the majority of these disorders, we don't have adequate treatment. And the big question, of course, is why, especially given a wealth of knowledge, decades of neuroscience research that have identified proteins, for example, that are either missing or dysregulated and causative of disease. And this has really led to the rise of protein biologics as a therapeutic modality, especially in the periphery. So you could simply produce protein in a dish and either provide replacement or supplementation to patients. It avails us to previously undruggable targets with improved safety profiles because you can provide the native protein as your therapeutic. It is, of course, expensive to manufacture proteins in a dish, but nevertheless, protein biologics are actually on the rise in terms of percent FDA approvals compared to small molecule compounds. And so at CereCura, we're very interested in harnessing these decades of preclinical data sets to deliver therapeutic proteins to the CNS. And so, the big hurdle to delivering proteins to the CNS, of course, is the protective blood-brain barrier. So this is a multicellular barrier that ensheaths every vessel in your brain. And it's there to control the passage of molecules to the brain compartment. So by permitting glucose and other energy substrates, for example, and restricting potentially neurotoxic targets. And this means that it also restricts protein biologic macromolecules. And so this has been a significant hurdle for delivering proteins to the brain. And so you could, of course, just inject them, proteins that is, directly into the brain compartment. The challenge there is that proteins typically have short half-lives, lasting for about hours in terms of elimination half-life, whereas what you would like is extended treatment or production of protein in that compartment for days to weeks. And at CereCura, we're harnessing the versatility of lipid nanoparticle and mRNA technology that Umar so nicely described to, instead of deliver protein biologics to the brain directly, we can harness resident cells inside the brain to manufacture these proteins in situ. And so by doing this, we're quite confident that we can achieve brain-wide distribution of these therapeutic proteins for a sustained duration. And by simply swapping different mRNA cargoes in brain-optimized lipid nanoparticle carriers, we can produce virtually any protein that we want. And at CereCura, we're very focused on secreted proteins. So when I started in Brian McVicker's lab as a postdoc just about 10 years ago, it was actually the first lab or the first collaboration between Brian and Peter's lab to deliver nucleic acid payloads with what we consider to be a modern lipid nanoparticle. And this was to develop siRNA against a putative chloride channel that was thought to be responsible in brain swelling. But our research group has since pivoted. So instead of knocking down channels or other proteins, we're using mRNA to produce secreted proteins that we think have therapeutic potential. And so we can modify these constructs in multiple ways. And so as Umar nicely described, the LNP is comprised of various parts. And so we focus on optimizing the lipid nanoparticle to specifically target brain cells. And you can do this in various ways by modifying the different components. But you can also modulate the mRNA sequence as well. And so in addition to just swapping out the coding sequence to encode for different proteins, you can also engineer it for, say, by changing the signal sequence to improve protein secretion, changing the UTRs to improve mRNA translation efficiency or even durability. And the advantage of this, of course, of this platform technology is that it's, of course, validated in the billions of us that received our COVID vaccine, our COVID mRNA vaccines. It's scalable, so the manufacturing process is a physical one, and it's very cost effective. And then, of course, as I mentioned, and as Umar mentioned, it's plug and play, so you can easily swap out the mRNA to code for whatever you want. And in our case, Umar very nicely illustrated the problem with lipid nanoparticles in that, especially when administered intravenously, they're highly liver tropic. But when you inject them in the brain, at least with the LNPs that we're developing, we get very nice broad distribution in the brain with specific uptake predominantly in glial cells. So this is an example of a mouse brain that's been injected with LNP mRNA encoding for mCherry, which is a red fluorescent protein. We get very nice distribution in the brain, specifically along white matter, and we seem to get the majority of our expression in oligodendrocytes and astrocytes, which are glial cells, also microglia. And so we're taking advantage of this glial specificity to convert these cells into our bioreactors, so in our protein manufacturing plants. And so we like glial cells for this job because they're first the most abundant cells in the brain and they're ubiquitous. They're metabolically flexible. They provide a lot of metabolic support to neurons. Unlike neurons, they replenish, and it allows us to use these cells as a disease agnostic platform. And so when we swap out the mRNA from mCherry to other mRNA sequences encoding for secreted proteins, this is de-identified, but this is FGF21 and EPO on the left here. We can get a nice expression of either for, and I'm not showing that, but several days post-injection. One of the typical criticisms of lipid nanoparticles is that there is immunogenicity, and we certainly do see that in the brain. However, it's completely manageable and highly transient. So we see a small increase after 3 hours that typically subsides within 24 to, and definitely by 48 hours. indicating that the majority of this response is from the lipid nanoparticle because we're still producing protein and we know the mRNA is still there. But we can completely ablate this by a pre-dose, an IP dose of dexamethasone, which is actually standard of care for LNP siRNA-based medicines. And so we're using this as a platform to produce various therapeutic proteins to target different indications. So you can, say, produce enzymes in a metabolic disorder. You can produce growth factors to promote neuroregeneration after trauma, cytokines for immunotherapy and brain cancer, and so on. And so as our first proof of concept for this platform in the brain, we are focusing on rare disease, specifically on a rare disease called neuronopathic Gaucher. And so this is a rare lysosomal storage disorder. It's one of 50 neuronopathic lysosomal disorders that's caused by mutations in the GBA1 gene. And so this is a vital lysosomal enzyme that breaks down glucosylceramide in the lysosome. It's interesting to us because type 1 is a peripheral disorder that is effectively treated by recombinant enzyme replacement therapy called CereCura. It's given biweekly, and patients can live effectively a normal life. But CereCura doesn't cross the blood-brain barrier, and so patients with types 2 or 3 or the neuronopathic variants are left with no treatment option options whatsoever, and these are fatal disorders. And so we're using LNP mRNA to produce GBA1. So that's the gene. The actual protein is glucose riboside or GCase, where we can produce GCase in astrocytes and oligodendrocytes. It's secreted and then endocytosed by neural cells by a mechanism of cross-correction, which is a well-characterized physiological pathway in the field. But this gene is also important for Parkinson's. And so mutations in GBA1 are actually the leading risk factor for Parkinson's disease. And when you mutate GBA1, you get accumulation of the pathological peptide alpha-synuclein. And it's thought to be because glucose riboside is responsible, at least indirectly, in the clearance of alpha-synuclein from cells, specifically dopaminergic cells. And so we develop our LNP mRNA therapeutics in a number of ways. And so this is an example in collaboration with Dr. Håkon Nygaard at UBC, who's developed a very nice human-based iPSC-derived neurosphere model where we can screen our lipid nanoparticles in a high-throughput way. This is just an example of a time course of LNP mRNA encoding for mCherry and the expression afterwards, where we can see very robust expression within a few hours and peaking over 24 to 48 hours. And it can be actually quite long lasting in these models. And so we can use this to actually engineer and screen our mRNA constructs. And so this is just a sanity check where we can demonstrate that we can produce glucose riboside, and we're seeing it both in new ends, so nerve cells, as well as GFAP positive astrocytes. We can demonstrate that the secreted GCase is active, but also that it is being endocytosed in untransfected cells. And so if we take supernatant containing secreted GCase and then apply it to untransfected spheres, we get very nice localization in nerve cells as well as lysosomes, so that it's achieving cross-correction as intended. And then when we inject it in vivo, so this is actually an example in rats. So we've done this in both mice and rats. We get very robust expression in the brain. So this is a zoomed in example, again, where we can see very nice expression in the corpus callosum, which is what I showed previously. But when we look at other brain structures, for instance, the striatum, which is highly relevant in Parkinson's, that's where the dopaminergic axons send their axon projections to synapse. And that's typically where the terminals start dying in Parkinson's disease. We can get very nice mCherry expression, again, predominantly in glia, so mostly in OLIG2 positive oligodendrocytes, but also astrocytes, and very rarely nerve cells. So even if we think we see some positive nerve cells, they're typically just a neighbor to a glial cell. We know we can produce GCase quite robustly, and so this is an example of, so on the right, of immunostaining for a flag-tagged GCase, again, showing really robust expression in the striations. And despite not seeing any evidence of LNP transfection in nerve cells, we can get a GCase flag signal in the lysosomes of these nerve cells in the striatum, indicating that we are achieving cross-correction in vivo as well. We can use this as a platform as well to screen for our different LNP formulations, and so we can test different LNP formulations that we tweak in various ways. So either by tweaking the ionizable lipid or the helper lipid to see which LNPs have the most activity in the brain. And the profiles are often very different. Sometimes we get activity leading to expression very early that tapers off very quickly, whereas some of them can lead to more prolonged expression up to 7 days or even more. And importantly, when we do this, again, this is with the direct brain injection approach, we do see our GCase protein everywhere. So everything that we produce here is human GCase, and our antibodies really don't detect the native mouse protein. And we get very robust expression in various brain structures like the corpus callosum, the cortex, the striatum, the hippocampus. But even in deeper brain structures like the midbrain and cerebellum, much higher than background. And so we're moving into more translational models now to develop this as a platform to treat human patients. And so in a collaboration with Dr. Mark Zembrowski here at UBC, who gets fresh surgically resected human brain tissue. These are from patients with intractable epilepsies. And you can actually section these tissues and then culture them. So it's effectively an organotypic slice culture system. We can use this to screen LNP activity again and just demonstrate that we can even use this as a platform in human tissues. And so when we use this to produce erythropoietin, for example, as a reporter with different LNP formulations, we can get very nice, robust expression over several days of this protein from the supernatant. We can confirm, again, using LNP cherry, that the types of brain cells that we're transfecting are consistent with what we're seeing in the mouse, and it does appear to be mostly glial cells. And importantly, we can use this to produce our therapeutic proteins of interest. And so this is examples of producing GCase using 2 different LNPs. And over about the course of a week, we can see production and secretion of this protein. So again, this is GCase from the supernatant at about a 4-fold increase from the physiological levels. And what's interesting about this too is, especially for neuronopathic Gaucher disease, this is far and above levels that you would need to reach to restore enzyme activity. And so typically, even about 10% can be good enough to reduce the pathological substrates. And so at CereCura, I mean, we certainly use lipid nanoparticles to deliver our mRNA cargoes. We certainly are more of a cargo developer, and we collaborate with partners to test various LNP formulations, and especially targeted LNPs and with Umar and co at the NRC. And so what is, of course, of great interest to anyone studying the brain and developing drugs for the brain is to have a BBB crossing LNP, which Umar summarized very nicely in his talk. And so this is really the silver bullet for neurology. And there are multiple delivery options that have been proposed across the BBB. Receptor-mediated transcytosis is the most common one when considering lipid nanoparticle cargoes and mRNA, because I mean this is a pretty large nanoparticle, and so harnessing transcytosis pathways, for example, by transferrin receptors, you can putatively deliver your LNP mRNA within the BBB compartment. And so in collaboration with the NRC, this is to deliver EPO as a reporter. We have some very nice data showing that this is achievable. And so in the BBB LNPs, again, this is looking at EPO in various fluids and tissues. We get a nice increase in EPO in the brain CSF that we don't really see in plasma. And in whole brain lysates, we also see an increase in EPO expression, indicating that we are getting bona fide delivery of our LNP mRNA to the brain. And so in summary, I'll conclude by just saying that we think that we have a nice platform here to harness glial cells as a plug-and-play approach to deliver protein biologic to the CNS. For the most part, we use direct CNS applications or administrations as a proof of concept. But by doing this, we can achieve brain-wide distribution of protein, and we can continue to test various LNP formulations, specifically targeted LNPs, to see if we can do this using intravenous administration. We can use this approach to produce therapeutic concentrations of GBA1 or GCase, and we're currently testing that in disease models now, including with brain-targeted BBB crossing LNPs. And we're using this as a therapeutic platform for neurological disorders at large. So we also have programs in Alzheimer's and spinal cord injury, the latter where we can use a different delivery approach, for example, going intrathecal in the spinal cord. And so I would like to acknowledge everyone that was involved in this work. So the CereCura team, our co-founders are LP Bernier, myself, Dr. Peter Cullis, and Brian McVicker here at UBC. We also have a fantastic team of scientists, both neuroscientists and mRNA biochemists who do all this work. And I would also like to acknowledge our collaborators at UBC, Hakon Nygaard and Mark Sambrowski specifically, as well as at NRC, Umar, Naz, and Brian contributed significantly to the LNP formulations for this work. And of course, thank you to our funders and especially NRC for funding a lot of this. So thank you very much.
No, thank you, Nick. That was a very, very excellent talk. So I guess maybe Scott, we're looking at potential questions.
Yeah, I think we have a couple questions. I will say just thank you to everyone. I think just really fantastic to hear from all sorts of people across the innovation ecosystem. Universities and research institutes, Canadian Blood Services, and of course, commercial development. I think all of that's so important to really bring this forward. So it's great. And apologies for going a little over on time, but we will stay on to do the questions. So I saw there's a couple. Maybe the first one I see in the chat from John Prietel was for Umar, and he was asking about whether we have data on the immunogenicity or immune response against the single domain antibodies when we use them in LNP. I don't know if you have anything to say about that, Umar.
Yes. These antibodies are from camel and humanized. So they are foreign to mice, like immunocompetent mice. But generally, our testing has been just short term. So we don't really look at longer term scenarios. But again, at the same time, single domain antibodies have been approved in human and tested, and I think have a good safety profile. Again, it really depends on, there could be, if a sequence on it is, triggers the immune system, it's possible, but I don't think it's case by case from my understanding. So I don't know if that's...
Yeah. So I would say kind of generally single domain antibody immunogenicity is a question we get a lot. And of course that applies also in our CAR T study. Whether it's an LNP or a CAR T or injectable protein, you can get antibody responses against those targets. And so far, I would say for the data that's been published, anti-drug antibodies do occur against single domain antibodies. It seems to be variable depending on the particular properties of your single domain antibody, not something that's unique to single domain antibodies. Some are more hydrophobic or hydrophilic or oligomerizing. There's different properties that can enhance their kind of immunogenicity profile. So for instance, there's a CAR T product for myeloma that's based on single domain antibodies derived from llama. And they have reported anti-drug antibodies in that case. That's not our our CD22 CAR T. That's a BCMA CAR T called cilta-cel. And in that case, anti-drug antibodies occur, but don't seem to interfere with the response. So I think similarly in the LNP, if we're going to inject multiple times with a single domain targeted LNP, we may have immunogenicity, but it may or may not interfere with the activity. So I think it's something really to be determined and something we'll track, I think, as more of this gets into the clinic and something actually we intend to also look at in the CD22 CAR T trial. I saw there's another question in the Q&A about in vivo CAR T. And I think this is a really interesting area. Of course, I think you touched on it a bit, Umar, as well. I think it is a really important area, a lot of investment and interest in that area, rather than needing to remove cells and make a CAR T ex vivo, which is a very expensive and complex process. Can we inject a viral vector or LNP or maybe other strategies to modify cells in vivo? That's something we're very interested in and working on. And I think it comes down to understanding your product. So I don't know if anyone wants to comment on that. Umar, do you have anything to add?
No, I mean, I think you say some years ago it would be a bit more far-fetched, but all of a sudden, I think people are there are some positive data, and companies are progressing in that space. I think like we do have tools that could be very effective in that space, but it's a matter of us exploiting it with the right application. So maybe like autoimmune disease or transplant is definitely a viable space. I think that where you need to modulate, but not like suppress too long necessarily. But again, we still to be determined for us in that space.
And maybe I'll just mention that I had said we're collaborating with for-profit commercial entities, to look at development of the CD22 CAR T in particular. Nanotherapeutics is an example of a company we're working with to see if LNP delivery of our CD22 CAR could be a strategy. And of course, we're looking forward to move that quickly towards development. And hopefully that can be a solution to really make this more broadly accessible, because accessibility and affordability was always the centerpiece of the CGT program, and I think that's still an ongoing process. I think we'll see these technologies develop over time. So good question, Nazanin. I see we have a hand up. Teffer, do you need to let Robert maybe turn on the camera and mic, please? Happy to hear your question, Robert.
Yeah, thank you. Oh, I can get the camera too.
Yeah,
Robert Merson. So I'm a consultant with CCRM and Omnia Bio. Really excited about the CAR presentations. We're very familiar with the Click team and all the work you're doing. I think that's exciting. My question, or almost more of a comment, is going to be for Dr. Lee. We just are finishing up actually currently, but we had a poster at the ISCT meeting in Dublin on a CAR NK manufacturing platform. So this is one we've been developing over a couple of years at CCRM, actually with Cytiva as a partner, transferring to Omnia Bio for that capability. But we do not have a CAR NK client. We are doing work on CAR T now at Omnia Bio, manufacturing CAR T. But 3 years ago, we thought this was going to be a really exciting area and a growth area, but we haven't identified a project on that yet. So really just a comment to say we were excited to learn about what you're doing. I really enjoyed this Made in Canada theme, but then I saw you're working with the South Koreans on your manufacturing process development. So just wanted to let you know that there's now a domestic option for NK manufacturing as well.
So there is anyway, it's very disappointing you don't have like a good customer for your CAR AI. But the thing is, I think it's really those in the market is the up and down, and then also sometimes science the driven, right? So I think it's we just waiting for because there's a CAR-NK product and a CAR T-cell product, there's pros and cons, and then what we can really highlight, this is, I think, is what we kind of waiting, to see in the new science paper, and then if a scientific paper really show dramatic response, I think we can really attract the market to the CAR-NK as well. Yeah.
Let's hope so. Thank you for letting me ask.
Okay.
Thanks for your question, Robert. It's this webinar, we wanted to hope to make those types of network connections. So we're really happy to hear from you at CCRM and Omnia.
Awesome. Thank you.
Do you mind if I can contact you later on?
Yes, please.
Yeah.
I was sort of looking for. I can put my email in the chat.
Yes, please.
Maybe that's the easiest.
Thank you.
Thank you. Yeah.
There is a question from Brad there too in the Q&A.
Right. I'll read the question maybe if you like, Brad. Thank you, everyone. Brad, do you think the G2R system could potentially be adapted for CAR Treg-based approaches in autoimmune diseases or transplantation? Brad, please.
Yeah, thanks for the question. And absolutely, anytime you need an IL-2 signal, we see application here. And it's quite exciting to think about Tregs, because if somebody's making a CAR Treg, it means that they're trying to suppress something. So it's either a solid organ transplant, for example. And their current CAR protocols are very problematic. Like even the lymphodepletion is something you don't, clinicians are really wary of doing in an autoimmune or transplant setting, because you lymphodeplete and then you get the homeostatic proliferation that follows that, and that could potentially exacerbate the very condition you're trying to suppress. And so with our G2R technology, the hope would be, we haven't proven this, far from it yet at this point, but hope would be that you could avoid lymphodepletion entirely and just put your Treg product in, give some GCSF, and get a good Treg effect that way.
Great. We have another question about sex and ethnocultural variants and how that's studied in this context. Maybe I'll, to spread around the questions. Maybe Harry, I'm sure that's something you've considered in your stem cell work. How are you kind of looking at those sort of variants as far as either in your gene therapy development or maybe in your stem cell work? Maybe you want to comment on that.
Yeah. So. We do notice differences from age is a critical factor. In some of our other work, we're finding that sex does play a role, especially in certain immune conditions, immune cell scenarios. What we generally try to do is we try to increase our N numbers as high as we can, and that's definitely a very important criteria that needs to be considered. And actually at Canadian Blood Services, in our new research and innovation strategy, one of the things we're trying to look at is in all the different blood products we're providing, we're taking into account genetics, metabolomics, to understand what is the impact of and sex, of course, and gender. How does this impact the efficacy in the people receiving it? So, yeah, unfortunately, research funding is not enough to do those large GWAS studies. You need thousands of donors, but we are definitely taking that into account as much as we can.
Yeah, definitely ethnocultural, I think, is a factor in the sickle cell disease, as you mentioned, Harry. Obviously, a disease predominant in African populations, I think. Rossini, you've got your hand up.
Yeah, I have a quick question for Harry. Given that, with the CGD program, one of our main goals is really making these therapies accessible to a broader patient population, and accessibility also goes hand in hand with affordability. But so I was wondering the approaches that you're looking at for editing HSCs. How do you think that the cost of goods would compare, and then obviously, that leads to pricing for something like Casgevy.
Yeah, so I mean, so no one's actually done this specifically in the context of stem cells, but a clear example is this work out of Alberta done in the context of CAR T, where they've come out and they said if if it was done by a non-for-profit situation, the product CAR T would probably cost around 50,000, is what the study said. I mean, it's a not published abstract. Again, I'm citing someone else's work. So all I'm trying to say is that they are going to be significant. If I were to venture a guess, there would be differences in the cost. But I think what the CGT program really, my personal opinion, has helped us is we're not just creating an alternative product, but the idea is that it's going to be a better product. And that's really what's really exciting. And even if we look at the click trial results, the product that's made in Canada it's a really good product. And that's really something we want to focus on, and what Scott's work's doing in clinical trials too. Hopefully the goal is it's not just another product, but it's a made in Canada better product that will help us Canadians. And yeah.
Thank you.
Umar.
Yes, just actually a question more for Nick. Related to the is also related to the question in the Q&A, but the non-viral manufacturing. Maybe what Canada is doing. I mean, you're part of a a center that has a lot of non-viral work, and also a big vision to, I guess, extend that in Canada. So I don't know if you have any insight or comments on how that's growing, and how you guys foresee that kind of manufacturing part of it in Canada.
I mean, at least in the Vancouver ecosystem, there's a big push to be an international hub for LNP mRNA drug development. And so here at UBC, there's been a number of different initiatives. There's going to be a new facility coming up, both here at UBC campus, but also at the new St. Paul's Hospital, all for the development of personalized medicines, as well as LNP mRNA. So all non-viral, which is, or at least the majority of it non-viral, which is very exciting. And it's fortuitous timing for us. And there's certainly a lot of other CDMOs in Canada as well that are popping up to scale for GMP level LNP manufacturing. And so, I think it's a good time for LNPs, and specifically for us, one of the, at least for now, if we don't have to coat the LNP corona with a targeting moiety, I mean, it does simplify the manufacturing process, but this might be something that you can better speak to for manufacturing targeted LNPs. But at least for a lot of conventional LNPs, that's really the key advantage over viral delivery, is that it's very straightforward. It's just mixing 4 ingredients and putting them in a tea mixer at the simplest level.
Maybe to sort of follow up on the sort of broader picture, Nick, how do you feel about taking something like that to trial in Canada? Is that something on the radar for you? Do you see that as a challenge? Is capacity there to, do these first in human trials with LNP technology?
I think for us, it sort of depends on what the first indication is going to be. So, I mentioned that GBA1 disorders, neuronopathic Gaucher disease, but also Parkinson's, NGD is quite rare. So, I mean, in North America, there's probably hundreds of patients, so very few in Canada. And so that would be a challenge. But I think if it's GBA1 PD, certainly that's more realistic. And I think given the push at the academic research hospitals to have clinical trials hosted here in Canada, I think that would be feasible. And we're certainly exploring that.
I think, to make the point, though, that it does need to plug into a larger network, especially in these rare disease situations, I think is really well taken. So, all
certainly,
right.
All right, I think we, if there are no questions, sorry again, we've gone over time, but I think this has been fantastic presentations, fantastic discussions. Thank you very much to the speakers, to everybody who attended, and thank you to BioCanRx. So we have recorded this presentation, and BioCanRx has kindly agreed to put this on the internet, and so it will be available for you to browse through and listen again and share. I think we will try to also in there include a way to contact the NRC if you would like to collaborate, and if you're interested in any of these tools and technology platforms to use in your own research. Thank you again.
Thanks, everyone.
Thanks,
Thank you so
everyone.
much.
Thanks, everyone.
Thanks, everybody.